Hi everyone, welcome back. Today we are going to be discussing Chapter 17, which focuses on sex in the brain. So we're largely going to be talking about sex hormones and how they play a role in important biological mechanisms for reproduction, but also how sex hormones affect our mood, our cognition, and our behavior. So the drive to reproduce is very powerful. It's as strong as our motivation to eat or drink. Now, the brain hardwires sex for reproduction, and reproduction is highly regulated by several different systems, such as the subcortical structures. So, this includes the limbic system, which we're largely going to talk about in the next chapter when we talk about how the brain processes emotional memories. So, stay tuned for more on the limbic system. But also, reproduction is highly regulated through conscious control provided by the cerebral cortex. For the most part, the neural control of our sexual organs is going to use that same somatosensory and motor pathways that we've talked about in earlier chapters. So, for example, the hypothalamus, again, is going to be a key commander in helping regulate our sex hormones and when they're produced and secreted. So, how do we choose a partner? Interestingly, we are more attracted to people who are different from us. This may not surprise you. In order for us to maintain sexual reproduction, we need to select a partner that has good genes and a strong immune system. This is obviously going to increase genetic diversity and strengthen the fitness of our offspring. So, how do we typically select a partner? Interestingly, this isn't always a conscious effort. Today I'm going to be talking about these molecules. This is known as the major histocompatibility complex, or MHC. These MHC molecules are a crucial part of our genome with respect to the immune system. For example, MHC plays an important role in our scent, and it can allow us to actually detect if a potential partner is genetically different from ourselves. MHC molecules are commonly found on antigen-presenting cells. Now, we have two key immune systems. We have our innate immune system that we're born with that can help us and protect us against things like bacteria, but we also have an adaptive immune system. The adaptive immune system is a little more complex. It utilizes specific immune cells that are basically in charge of creating antibodies when we come into contact with a virus. These antibodies are stowed away, and this immunological memory actually protects us from the same viruses in the future. This is commonly utilized for the conceptualization of vaccines. So, for example, if you've had the COVID vaccine before, your body has produced antibodies to fight it if you ever come in contact with it again. MHC molecules, again, are presented on these antigen-presenting cells, and they're basically a key to our entire biodiversity. So it allows us to essentially detect if a partner is genetically different from ourselves. So what exactly does this mean? I'm going to talk about this more on the next slide. So sex has actually evolved to help us in pathogen resistance. So I'm going to use Bella and Edward from Twilight as a great example, which seems silly. But essentially, if you've ever read the Twilight books or watched the movies, you know that Edward always talks about his attraction towards Bella, that her blood specifically smells really good to him. It's like his own personal scent of perfume. It's like a drug to him. He's very attracted to her. Well, of course, hopefully, when you're choosing your partner, they won't be a vampire, but we can detect the smell of our partners and how differently their genetics are from our own through MHC ligand peptides. These MHC ligand peptides are essentially like our own personal perfume that reveals our MHC genetics. This is a way that we can subconsciously detect if our partner has different genes compared to our own. The partner we usually choose will typically have MHC alleles that are different from our own, and thus we have different antibodies to fight potential pathogens. So why is this important? This brings me to the Red Queen hypothesis, which is a key hypothesis utilized in evolutionary psychology. MHC-based mating is really important because it helps us find a partner that's genetically different from ourselves. And this is important for reproduction. If our partner is genetically different from ourselves, this is going to provide our offspring with greater genetic diversity, and it's going to strengthen and bolster their immune system and increase their fitness. This is going to help provide them with higher resistance to infection. This hypothesis comes from Alice in Wonderland. It takes all the running you can do to keep in the same place, essentially noting that we have evolved throughout time. And in order to stay in this same place, to survive, to be stronger and fit, we need to find partners that are more diverse from us so that our combination of genes can help our children fight off new potential pathogens. So we've evolved in the presence of pathogens. We come into contact with different bacteria and viruses all the time. So now we require new combinations of our genes for stronger resistance against these potential new pathogens. For example, the COVID virus. This is a new virus that we're coming into contact with, and we are going to evolve in the presence of COVID, and the combination of genes is going to help our offspring and their future offspring fight new viruses or pathogens that develop. So essentially, sexual reproduction has evolved to help our children increase their fitness and beat pathogens. And if you'd like to read more about this, I added this link to the paper by Malinsky and colleagues published in 2006. So now I'm going to give you a real-life example of how major histocompatibility complex molecules play a role in mating behavior and reproduction. So interestingly, in this study that was conducted in 1995, often known as the T-shirt study, this study was a really integral part in recognizing the importance of MHC molecules in partner choice. So I'm going to walk you through this study. First, this was conducted at a university, and male students were asked to wear a t-shirt for 2 consecutive nights while sleeping. So we could imagine if this was happening at Vanguard University, male students would be asked to wear pajama shirts, the same pajama shirt for 2 nights in a row, and then they would need to bring that shirt back to the researchers. So you can imagine how funny this study would be because female students were asked to come in and rate the odors of the 6 different t-shirts. And interestingly, women preferred the smell of men who had different MHC alleles from their own. As you can see here in the green box, these graphs right here are demonstrating women who were not on birth control pills, and I'll talk about the importance of that in a moment. So essentially, these women were not on a birth control pill, and they were asked to rate the pleasantness or intensity of male odors. And interestingly, males who had more dissimilar MHC alleles from their own, they found to smell more pleasant, which demonstrates that even though we may not realize it subconsciously, if we like the smell of our partner, it's probably because MHC is playing a role here, because our partner is more likely to have different MHC alleles from our own. And interestingly, in the second set of graphs I wanted to show you guys, birth control was associated with the opposite preference. So they actually looked to see if women were taking oral contraceptives. That's demonstrated in these graphs right here with the black bars. And interestingly, if women were on the birth control pill, this actually was associated with that opposite preference, in which women actually had an increased preference for the smell of men who actually had more similar MHC alleles to their own, which is really interesting. And I'll talk a little bit more in today's lecture about how oral contraceptives can affect not only reproductive behavior, but even cognition and mood. Essentially, this study was integral in suggesting that MHC or linked genes specifically can influence our human mate choice. And so I find this study fascinating, and a lot of my students usually do. And I always joke that if you want to go look for a boyfriend or girlfriend, you just have to go around campus and smell them out. So now we're going to talk about the genetics of sex. So we're going to start talking about chromosomes and the importance of chromosomes during development. So in order to talk about the genetics of sex, I'm going to reintroduce chromosomes, which we did talk about in the earlier chapters of this class. Now remember, DNA is super long. We have this long strand of nucleic acids that are going to code for different genes. We have the same exact DNA present in all of our cells in our body. Now, chromosomes are the piece of DNA comprising a single gene, and they provide the unique information needed to construct a particular protein. If you remember, protein synthesis is that key endpoint of gene expression, right? We have about 25,000 different genes in the human genome, which is pretty incredible. And as you can see here in this image, this is showing us the human chromosomes. There are 23 pairs of chromosomes present. As previously mentioned, DNA is organized into 46 chromosomes, or 23 pairs. We receive 23 from our father and 23 from our mother. Each of us have two versions of the chromosomes 1 through 22. And as you can see in this image, they're conventionally numbered in order of decreasing size. As you can see right here, you can notice how much smaller the Y chromosome is compared to the X chromosome. Females are born with 2 X chromosomes. They receive one from each parent. Males have an X chromosome from the mother and a Y chromosome from the father. These are called genotypes, and genotypes specify a person's genetic sex. Now I'm going to provide you with some examples of what can potentially go wrong if there's a defective gene on an X chromosome. A defective gene on an X chromosome may result in something called an X-linked disease. And I'm going to give you some examples of X-linked diseases in a few moments. But first, I want to give us some context. So let's say a female has a defective gene on an X chromosome. She may not experience any consequences of this if the gene present on her other X chromosome is normal. But think about this in males. Males only have one X chromosome and one Y chromosome. So any defect in that single X chromosome of a male can potentially lead to a developmental defect. And so this defect on that single X chromosome in a male is often known as an X-linked disease. So a great example of this is actually red-green color blindness. Red-green color blindness is usually induced by a defective gene on the X chromosome, and that's why it's more common in males. Interestingly, another X-linked disease that's less common is hemophilia, and this is a disease in which the blood does not clot properly. So people who have hemophilia, if they are severely injured, this can be very dangerous. You don't want them to bleed out, so you'd have to get them to a hospital as quickly as possible. Although this disease is more rare and it is treatable, it is an X-linked disease, meaning it's usually induced by this defective gene on the male's single X chromosome. Now I'm going to provide you with some examples of chromosomal disorders. So this is when a person maybe is born with one extra chromosome or born without a chromosome. The first example I'm going to share is called Klinefelter syndrome. The genotype for Klinefelter's is XXY. Now I removed the textbook images that provide the examples of these different syndromes I'm going to discuss. So if you'd like to look at the images, you can go back to your textbook. But essentially, this is when a person is born with an extra X chromosome. So this is when a male would have the genotype XXY. And due to that extra X chromosome, that can induce infertility in the male. And other symptoms include a lower IQ and impaired cognition. The second sex chromosome abnormality I'm going to discuss is Turner syndrome. Again, if you want to see images with examples of Turner syndrome, you can access that in your textbook. The genotype for Turner syndrome is XO genotype. And this is when a female is either born with just one X chromosome or a partial or absent second X chromosome. So this is actually affects about 1 in 2,500 female births, so it's not totally uncommon. And essentially, the babies who survive this have a wide variety of phenotypical characteristics, including short stature, a receding jaw, a webbed neck, and visuospatial and memory difficulties. Also, during development, females who are born with Turner syndrome, their ovaries are abnormal. So especially important during adolescence, estrogen replacement therapy is a key therapeutic strategy. And we're going to talk about how hormone replacement therapies are important. If you remember, we've talked about this previously in the context of John F. Kennedy. If you remember, he had Addison's disease and could not develop cortisol properly, so he had to receive cortisol replacement therapy. Similarly here, if someone is born with Turner syndrome, they can receive estrogen replacement therapy to help with normal breast development and maybe increase likelihood of menstruation. So presumably, because the loss of an X chromosome in a male is lethal, there are no known individuals with this YO genotype. It's physically impossible for a male to live without the X chromosome. Now we're going to talk about the development of reproductive organs. In previous chapters, I talked about the importance of the development of the central nervous system and the peripheral nervous system. We spent a lot of time talking about neurulation and how the neural tube has to develop properly to develop the brain and the spine. Similarly, we're going to talk about the beginning of the reproductive system from conception and beyond. So when we are conceived, during those first 6 weeks post-conception, our sexual organs are essentially undifferentiated. So we start development with both Müllerian and Wolffian ducts. Now, Müllerian ducts will end up inducing the female reproductive system, and Wolffian ducts will end up developing into the male reproductive system. And I will talk more about that on the next slide. But interestingly, because the body is undifferentiated before that 6 week of development, there are certain genes that need to be switched on to activate the development of the Wolffian ducts and the formation of the male sex specifically. So let's talk about this in a real life example. So males are born, of course, with a Y chromosome. And the Y chromosome actually contains a gene called the sex determining region of the Y chromosome. We'll refer to this gene as SRY. This SRY gene actually codes for a protein and transcription factor called testes determining factor, or TDF. So remember, a transcription factor is going to increase the likelihood of a gene being expressed, and that final result of gene expression is protein synthesis. So ultimately, this gene that's present on the Y chromosome is necessary for the development of a male, because this is going to induce the development of the Wolffian ducts, and it is going to essentially degenerate those Müllerian ducts that first started developing before that 6 week of development. In a human with a Y chromosome, if a human is born with a Y chromosome, obviously their genetic sex is male, and that SRY gene develops as a male. Without it, the body will just continue to develop into the female sex. As I previously mentioned, from conception until about week 6 of development, a baby is sexually undifferentiated and starts developing both the Müllerian and Wolffian ducts. It isn't until about week 7 or 8 where testes determining factor, or TDF, is expressed, which will determine whether a person becomes male or female. So image A is showing us the undifferentiated reproductive system with both the Müllerian and Wolffian ducts. In image B, this is showing the male sex. So this would be occurring if the SRY gene was present on the Y chromosome, which would increase TDF expression. TDF expression induces the development of the Wolffian ducts and the degeneration of the Müllerian ducts. However, if there is no SRY gene and no TDF present, The Mullerian duct is going to continue developing into the female reproductive organs, such as the ovaries and the uterus, whereas the Wolffian duct is going to start to degenerate. Now let's talk about sex hormones. First, we're going to talk about how sex hormones are synthesized and how they affect the brain and body. Sex hormones are steroids, and the one steroid we've talked about in depth in this class is the stress steroid cortisol. Steroids are synthesized from cholesterol, specifically HDL, known as high-density lipoprotein. Remember, we produce two different types of cholesterol, including HDL, high-density lipoprotein, and LDL, low-density lipoprotein. HDL is what we kind of refer to as our good cholesterol that you're going to gain from good fat sources. These are fat sources that include monounsaturated fatty acids and polyunsaturated fatty acids. So if you eat these good fats, which are commonly found in foods like avocado, olive oil, fish, this can help us increase our HDL. And this is really important to have in our diet as steroids are synthesized from cholesterol. Males and females both have androgens and estrogens. Testosterone is an androgen, and estradiol is an estrogen. All of our sex hormones develop from cholesterol, and cholesterol is synthesized into progesterone. Progesterone is then synthesized into testosterone, and there are two important enzymes that play a role in the creation of estrogen and DHT. In order for testosterone to form into estrogen, or estradiol, the enzyme aromatase has to convert testosterone into this hormone. The enzyme 5-alpha reductase is going to catalyze testosterone into DHT, which is another form of testosterone. Interestingly, our sex steroid hormones don't only play an important role in our reproductive behavior, but they can act as transcription factors to turn on gene transcription and translation. Thus, they can influence gene expression both in the brain and the body. Later on, I'm going to talk about how hormones actually play a really important role in learning and memory. For example, estradiol needs to be present during learning in rats. So we're going to talk about these experiments and how estradiol and estrogen could potentially increase the gene expression of these good, helpful genes that are involved in learning and memory processes, like brain-derived neurotrophic factor, or BDNF. We have several sex steroid receptors in the brain. This image is from the textbook, and this is demonstrating sex steroid receptors that are present in the rat brain. As you probably guessed, there is a high density of these sex steroid receptors in the hypothalamus and the pituitary. As we know, the hypothalamus is our captain, guiding sex hormone release from the anterior pituitary, and these hormones are then going to go to the gonads, of course. There are other areas of the brain that also have larger populations of sex steroid receptors. And in our next chapter that we're going to cover next week, I'm going to talk about emotional processing and emotional memory in the brain. And we will talk more about the importance of sex steroids in these emotion processing regions. I wanted to show you guys this image of a hypothalamus and pituitary gland. This is actually from a scanning electron micrograph of the pituitary gland from a monkey. And I think it's really amazing to see all of this beautiful architecture. We can see all of these blood vessels and veins that are coming from the parvocellular neurons in the paraventricular zone of the hypothalamus. That are going to allow those hypophysiotropic hormones to be released into the bloodstream that will then enter the anterior pituitary gland. And because the parvocellular neurons don't actually synapse into the anterior pituitary, as this is simply a gland, the main mode of communication between the hypothalamus and the anterior pituitary is via these hypophysiotropic hormones that are released into the bloodstream. So this is playing a really important role in that communication between the hypothalamus and the anterior pituitary. So as I noted before, the hypothalamus is largely playing a really important role in sex hormone release. We've seen images like this before when we were talking more about the posterior pituitary in chapter 15, but now we're going to focus on the anterior pituitary as it's important for releasing our important sex hormones that are going to go to the gonads. So parvocellular neurosecretory cells sit in the paraventricular zone of the hypothalamus, and they release these hypophysiotropic hormones into the bloodstream. The ones that I'm going to talk about today include luteinizing hormone releasing hormone, LHRH, or gonadotropin releasing hormone, GnRH. These two terms are synonymous and can be used interchangeably. So let's say that gonadotropin releasing hormone is released by these parvocellular neurosecretory cells. They're going to be dumped into the bloodstream from the hypothalamus, and they're going to travel down to the anterior pituitary. The anterior pituitary will receive these hormones from the blood, and this will stimulate the release of important gonadotropins. Gonadotropins are the hormones that are released by the anterior pituitary gland, and these include luteinizing hormone, or LH, and follicle-stimulating hormone, FSH. These hormones play an important role in testosterone and estrogen release, and follicle-stimulating hormone specifically plays an important role in sperm maturation and ovulation. When these hormones are released by the anterior pituitary, they're going to be transported via blood again, and they're going to go down to the gonads, and the gonads will then be able to release important sex hormones like testosterone and estrogen. So once again, the hypothalamus is the key captain of this ship, if you want to think about it like that. And even the hypothalamus is playing an important role in sex hormone release. Luteinizing hormone and follicle-stimulating hormone are involved in the menstrual cycle in females. Of course, we know this is important because luteinizing hormone and follicle-stimulating hormone help assist in estrogen release and the maturation of eggs. So let's look at the female menstrual cycle. In the top of this image, this is showing us the amount of gonadotropin releasing hormone. Remember, this is a hypophysiotropic hormone that's released by parvocellular neurons in the paraventricular zone of the hypothalamus. We're also going to see these blood levels of pituitary hormones that are released by the anterior pituitary, including luteinizing hormone and follicle-stimulating hormone. As you can see, throughout the female menstrual cycle, there are different fluctuations of hormones. This shouldn't be surprising, hopefully. But what's really interesting that I wanted to point out is during ovulation, the hypothalamus is releasing high levels of gonadotropin releasing hormone, and the anterior pituitary is releasing really high levels of luteinizing hormone, shown here in purple. There's also this spike in follicle stimulating hormone. And that's because these two important gonadotropins are playing a role in estrogen release and ovulation in the ovaries. So in addition to this first half of the cycle, estrogen levels are going to be higher, whereas in the second half of the cycle, progesterone levels are going to be higher. So ultimately, women's hormones do fluctuate. And just a quick review of hormonal control of sex. I'm bringing up this slide again because it's really important for your next exam. Know that cholesterol is converted to progesterone. Progesterone is converted to testosterone. And aromatase is this really important enzyme that converts testosterone into estradiol. Now we're going to talk about the bidirectional relationship between the brain and the gonads. This is known as the hypothalamic pituitary gonadal axis, or HPG axis. Now this probably sounds familiar because we've talked about the hypothalamic anterior pituitary adrenal medulla axis, known as the HPA axis, in the context of stress. Don't confuse these two because the HPG axis is playing a really important role in our reproductive behavior and sex hormone production. So let's take a closer look at the HPG axis. First, I want to note that the hypothalamus is actually influenced by psychological factors and sensory information like light responses from the retina. When neural input is received from the retina and sent to the hypothalamus, this can cause changes in the release of gonadotropin-releasing hormone from parvocellular neurons in the hypothalamus. And this is based on daily variations in light level. So let's quickly review. How does the hypothalamus regulate and direct our sex hormones? This is via the HPG axis. GnRH is released by the hypothalamus. Remember that this is a hypophysiotropic hormone that's released by parvocellular cells in the paraventricular zone of the hypothalamus. GnRH is dumped into the bloodstream and will go down to the anterior pituitary. This regulates gonadotropins, such as luteinizing hormone and follicle-stimulating hormone that are released from the anterior pituitary. And these gonadotropins are going to have a direct effect on the secretion of testosterone in the testes and estradiol in the ovaries. And the main route for these gonadotropins from the anterior pituitary is via the bloodstream to the gonads. And this is a bidirectional relationship. So reproductive organs are constantly communicating with the hypothalamus and vice versa. So as I noted previously, light received from the retina can actually affect gonadotropin release. Now, in some non-human species, strong seasonal variations in reproductive behavior and gonadotropin secretion occur. And this is largely because light inhibits melatonin production. And melatonin inhibits gonadotropin release. So day length can directly influence reproduction, and this is a biological and adaptive mechanism to increase offspring survivability. Interestingly, a pineal tumor could potentially inhibit gonadotropin release due to this hyperproduction of melatonin. And you can see that having a pineal tumor could render some serious consequences. Now, let's look at this in the context of seasonal breeding in sheep. So, as we can see during the summer and fall, we have increased daylight in the summer and early fall, and then the amount of light during the day slowly decreases. Breeding is largely going to occur during the fall, and this is an adaptive mechanism so that sheep can continue pregnancy throughout the winter. Now, you can think about this from an evolutionary perspective. If sheep were to give birth in the winter, that could increase the risk of their offspring dying. So really, this seasonal breeding is going to help increase the survivability of offspring. Now, as increasing day length occurs in the spring and summer, sheep will give birth and go back into their ovulation cycle. So ultimately, it is day length that is playing an important role in gonadotropin release during the fall to ensure that sheep will become pregnant in the winter and give birth in the spring when it's warmer, and this will ultimately increase the survival of their babies. So it's important to note that the peripheral nervous system plays a really important role in the sexual response. So sex is a coordinated effort between our parasympathetic nervous system, shown here in green, and our sympathetic nervous system, shown here in blue. Now, if you remember, we've looked at this image before, and we can see that all of these neurons that are innervating these different organs here in the parasympathetic system are largely secreting acetylcholine. Whereas over here we see that both acetylcholine and norepinephrine are playing an important role in stimulating different responses in different organs. So remember that the parasympathetic nervous system, this is our nervous system that we refer to as our rest and digest system, is actually responsible for increasing blood flow to the genitals during sexual intercourse. And the sympathetic nervous system is playing an important role in ejaculation. As you can see here, there's a coordinated effort between these neurons that are releasing norepinephrine and acetylcholine here during sexual intercourse. Now let's focus on the parasympathetic nervous system and how it plays an important role in neural control of reproductive organs. So in this image here, this is showing us two different areas of the spinal cord, the lumbar and the sacral regions. And so axons of sensory neurons, so sensory neurons here are shown in green, are going to head to the sacral spinal nerves. Parasympathetic pathways are going to become activated, and this increases blood flow to the genitals. This is induced by acetylcholine and nitric oxide. So these parasympathetic fibers are releasing acetylcholine and nitric oxide directly into the erectile tissues to help with relaxation and engorgement of reproductive organs by increasing blood flow. And so medications for erectile dysfunction, like Viagra, have actually been shown to enhance acetylcholine and nitric oxide mediated vasodilation here. So this is how the parasympathetic nervous system plays an important role in sexual intercourse. Now I'll talk about the sympathetic nervous system and how it plays an important role in reproductive organs. So again, let's take a look at the sensory pathways here in green. Sensory axons are going to synapse into both the thoracic and lumbar regions in the spinal cord. This excites the sympathetic neurons and coordinates a complex series of these muscle contractions that are going to play an important role in ejaculation and orgasm. And there are two different areas in the brain that are heavily involved in orgasms, including the medial temporal lobe and the basal forebrain. And when we start talking about brain mechanisms of emotion, we're going to talk about this a little bit more in depth. Now we're going to talk about mammalian mating strategies and how they vary between different types of mammals. Monogamy is not super popular across animal species, and so a more popular form of reproductive behavior among mammals is polygamy. This comes from the Greek word for many women, in which the male prefers to mate with many females. However, the female usually only mates with one male for each mating season, because obviously it is going to take that female a longer time to actually grow and give birth to the offspring. So polygynous mating, practiced by giraffes and most other mammals, has kind of this one-night stand quality to it, in which a male will actually never go back to check in on the outcome of his offspring and will usually not check back in with its mate. So this reproductive strategy is commonly seen in mammals, and it's seen in the specific species called the montane vole, which has been heavily studied in neuroscience. So we're going to talk about the neurochemistry and reproductive behavior of the montane vole. So voles are a part of the rodent species, and their pituitary hormones are largely regulating their reproductive behavior. So now I'm going to talk about two different species of voles. We're going to compare montane voles to prairie voles. So many neuroscientists have thoroughly studied these animals and their mating behavior. As you can see here, this is demonstrating that the voles are given 3 different chamber options after mating. They can select a partner chamber where they will stay with their partner after mating. They can be left in a neutral chamber where they can be by themselves, or they can choose to go to a stranger chamber, shown here in orange. Interestingly, these 2 different species of voles have very different reproductive behavior. So the montane vole, as you can see, prefers to sit in the neutral chamber, as demonstrated by this turquoise bar. In contrast, the prairie vole prefers to spend more time in this purple chamber with its partner after mating. So ultimately, it seems that prairie voles want to spend more time with their partners compared to the montane vole. Why is that? Well, it's because of oxytocin and ADH receptors. They're distributed much differently in montane and prairie voles. Now over here, these 2 images are showing us brain sections from a montane vole, and on the right, this is showing us coronal sections from a prairie vole. And the distribution of oxytocin and vasopressin receptors in their brains are demonstrated through these images. These are audio radiograms, and the regions with the highest receptor densities are the darkest. So if you see, the oxytocin receptors in the montane vole are very few and sparse. However, in the prairie vole, there are much denser amounts of oxytocin receptors. And based on what we know in class, oxytocin is that neurohormone released by the posterior pituitary that helps with bonding with your partner. And so thus, that makes sense that prairie voles would want to spend more time with their partner after mating because they bond with their partner due to oxytocin, whereas montane voles just simply have less oxytocin receptors. In monogamous rodents and primates, these oxytocin receptors are actually found to be elevated in brain areas that are associated with reward, like the ventral tegmental area. So this is very interesting that being maybe in a long-term relationship where you've bonded with your partner and you're in love with your partner is going to bring you happiness. And that's demonstrated by these oxytocin receptors that are found in these brain areas associated with reward. So if we look here at oxytocin receptors in the prairie vole versus the montane vole, we can see that there is a more dense amount of oxytocin receptors present in these brain areas associated with reward, such as the nucleus accumbens. If you remember in my last lecture, I talked about how the nucleus accumbens play a really important role in reward from drugs or food. So ultimately, bonding with a partner and being in love can actually increase oxytocin release in these areas of reward, which I just think is so cool. It's also interesting to note that different types of love are activated in different areas of the brain. What do I mean by this? Well, in imaging studies, demonstrating the differences between maternal and romantic love, we can see that these two different types of love are going to activate different areas of the brain. So, for example, in this study, a woman could be put into a PET scan. If you remember, a PET scan can allow us to look at activity and metabolism in the brain. And let's say that first, the researchers show her pictures of her children. This maternal love is going to activate these areas of the brain highlighted in yellow. Whereas if the researcher showed her a picture of her husband, her romantic love, these areas shown in red could be activated from that romantic love. So ultimately, we can see that red areas were more activated by these romantic partners or husbands. And thus, there are different areas of the brain that are playing a role in maternal love versus romantic love. And we're going to talk about this more in depth throughout the rest of this lecture. As I mentioned previously, montane and prairie voles have very different vasopressin and oxytocin receptors. So I wanted to investigate this further for you guys to see if this has been studied in humans. And so now we're going to talk about vasopressin gene variants and pair bonding in humans. And this has been studied. So scientists have investigated whether vasopressin gene variants could influence pair bonding in humans. So most of these studies are conducted via questionnaires and surveys where participants are asked to rate the quality of their marriage through these questionnaires. And the results demonstrate that in women specifically, there was no connection between a vasopressin gene variant and the quality of their marriage. However, men with a particular gene variant scored significantly lower on measures of the quality of their marriage and were twice as likely to report that a marital crisis had occurred in the year before the survey. And the wives of the men with the gene variant present also reported lower marriage quality than the wives of men without the variant. So ultimately, this study demonstrates that there is an association between this vasopressin gene variant and partner bonding. The function of the gene variant is still unknown, but these results largely suggest that even in humans, not just in voles, vasopressin receptors could play a role in pair bonding. So now let's talk about some neural areas that are activated in sex and bonding. Of course, we know the hypothalamus is activated as well as the posterior pituitary due to the neurohormone oxytocin. The ventral tegmental area and the nucleus accumbens are activated during sex, as these are areas of the brain that are associated with reward. This is like what we talked about in the last chapter, how food can actually facilitate dopamine release. Similarly, during sex, dopamine release is facilitated by oxytocin, and particularly in males, this can be compared to the response of a heroin injection. So this is a very powerful reward that's received by the ventral tegmental area and the nucleus accumbens. Additionally, sex has been shown to inhibit the prefrontal areas and the amygdala, which makes sense because the amygdala processes fear-based memories. And so particularly in women, the amygdala has been shown to be inhibited to reduce that fear response. The entorhinal cortex, which sits near the hippocampus, has also been shown to play an important role during sex. And we're going to talk about the entorhinal cortex in more depth later on, as it plays an important role in learning and memory, specifically emotional memory processing. Additionally, secondary visual cortices are activated, and the anterior cingulate cortex, the cingulate gyrus is shown right here, and the insula play an important role. Now, these are limbic areas that I haven't covered in too much depth yet in class. And on the next slide, I'm going to talk more about the insula specifically and how it's activated during sexual intercourse. The insula plays an important role in emotional processing and sexual desire. So here's the insular cortex. And so here in blue, we're looking at the anterior lobe of the insula, and in yellow here, we're looking at the posterior lobe of the insula. So interestingly, researchers have utilized PET scanning to understand which brain areas are activated during lust and love. So love would mean that the participant could potentially be looking at images of their long-term partner, like their husband or wife. So the posterior region of the insula is actually activated more during lust than love. So for example, this could be when you're looking at one of your favorite movie stars, maybe a movie star that you have a crush on. You don't know this person, so obviously you haven't bonded with them. You haven't released oxytocin in their presence. So this is going to be considered lust more than love. And this has been shown to activate the posterior lobe of the insula. Whereas the anterior parts of the insula are activated during love more than lust. So for example, if a participant was looking at a picture of their husband of 25 years, the anterior lobe of the insula would probably be activated because they've bonded with their partner. And this is largely due to oxytocin release. Interestingly, the ventral tegmental area is active in both sides in lust, and the right ventral tegmental area is activated in love. So remember, the ventral tegmental area, or VTA, is activated during reward. There's a large dopamine release that floods in this area for food reward, drug reward, and in sex reward. Now, the prefrontal cortex is involved in both lust and love. In the striatum specifically, which I'm going to talk about more on the next slide, is really interesting because we've seen that as lust transitions to love, it actually changes the way the striatum is activated. So let's take a closer look at this brain area. So in this study that I cited right here, you can take a look at it if you'd like. Researchers asked participants about their relationships and their happiness via surveys. And then they performed fMRI imaging to look at the brain. And so before I talk more about this, I just want to note that the dorsal striatum is located right here. And here we see this is the basal ganglia system and the putamen, right? So being in a romantic relationship was actually associated with greater happiness and reduced gray matter density within the right dorsal striatum. Thus, being in a romantic relationship is associated with positive experiences and a reduction of gray matter density in the right dorsal striatum, representing that there's actually a social reward that's activated within a romantic relationship. When you're with a long-term partner and you've bonded up with that partner, this romantic relationship actually changes brain tissue and neurochemistry, which I just think is absolutely amazing. So if you want to read more about the dorsal striatum and these changes following a romantic relationship, you can click on this link to read this paper that was published in 2016. Now I'm going to discuss the importance of estrogen on neuron function, in memory, and in disease. So estrogen's actually playing a really crucial role in all of these factors. Estrogen has a powerful effect on our neurons. It can affect the structure and the function of neurons. And by modulating the flow of potassium ions, estradiol, which is the primary form of estrogen, has been shown to actually depolarize neurons and induce action potentials. So in this study that was conducted at Columbia University, researchers found that estradiol treatment of tissue taken from the hypothalamus of newborn mice caused a great degree of neurite growth. So let's take a closer look at this study. In image A, this is showing us tissue from the hypothalamus of a newborn mouse that did not receive estradiol treatment, compared to image B, which is demonstrating the hippocampal tissue that was treated with estradiol. Estradiol actually increased neurite growth, which is pretty amazing because increased neurite growth plays an important role in learning and memory. So how does estradiol affect learning and memory? Well, estradiol plays a really important role in dendritic spine density. And if you remember at the beginning of this class, I talked about how dendritic spines play a crucial role in cognition. If you remember previously, I've shown an image of a child with cognitive impairment compared to a healthy child. And the healthy child had more dendritic spines and thicker dendritic spines compared to the child that was experiencing cognitive impairment. So ultimately, neuroscientists have also discovered that dendritic spines can fluctuate due to estradiol levels. And in female rats specifically, the number of spines in hippocampal neurons changed dramatically during the 5-day estrus cycle, which is similar to the human menstrual cycle. Now, spine density and estradiol levels peaked together, which is very interesting to see that if women maybe perhaps were producing a lot of estrogen at a particular time in their cycle, for example, during ovulation, there could be increased spine density during this time as well. And treatment with injected estradiol has also been shown to increase the number of spines in animals who have lower natural levels of estradiol. So ultimately, hormone therapy could increase dendritic spine density. And since spines are the major site for excitatory synapses on dendrites, as we've talked about dendrodendritic synapses at the beginning of this class, this provides a possible explanation for the fact that hippocampal excitability also seems to track the estrus cycle. Estradiol not only affects dendritic spine density, but it even affects the NMDA receptors that are present in the hippocampus. So remember, the NMDA receptor is the main glutamate receptor in the brain, and it's a key pathway for learning and memory processes. NMDA receptor antagonists have actually shown to decrease learning and memory. So we know that NMDA is facilitating this important memory consolidation. Peak in hippocampal spine number actually coincides with the rat's peak in fertility, so during ovulation, during the estrus cycle. During this period, a female actively seeks out a mate. So from an evolutionary perspective, if a rat was out in the wild, they would need heightened spatial ability to look for a mate. Similarly, even in a lab rat, they may require this heightened spatial ability that comes with a more excitable and NMDA receptor-filled hippocampus, because the NMDA receptor is necessary to learn these important, crucial spatial memories. So thus, the female rat brain actually fine-tunes itself on this 5-day estrus cycle in order to meet its changing reproductive needs. It's amazing that estradiol can affect the NMDA receptor in female rats. So how does estradiol increase the number of hippocampal spines and excitatory synapses? The detailed mechanisms are still not very clear, but we do know that estradiol increases the capacity for synaptic plasticity in the hippocampus. If you remember, synaptic plasticity is the ability for the brain to create new synapses, which are crucial for the neural mechanisms of learning and memory. So, synaptic plasticity is important, and we know that estradiol is playing an important role in increasing synapses in the brain. In the presence of estradiol, postsynaptic responses to glutamate are actually larger than they are without estradiol. And this causes synapses to strengthen. And we know that when synapses are strengthened, this can induce long-term learning and memory. This could also trigger an increase in the number of dendritic spines on pyramidal neurons that live in the hippocampus. So ultimately, having increased dendritic spine density on neurons in the hippocampus are going to facilitate this long-term memory and strengthen synapses. In rats, the hippocampus is very important for spatial memory and navigation skills. And in neuroscience research, we utilize several different behavioral paradigms to measure spatial memory. For example, the image here is showing the task called novel object recognition. This is when a rat is trained to decipher between two different objects. So for example, during training, there could be two objects placed in these locations in a chamber, and the rat is given maybe 10 minutes of uninterrupted movement to explore the chamber and understand where these objects are located. And then once the rat is put back in its home cage, there may be a 24-hour period for consolidation. And then the next day, the rat is brought into the same exact chamber, but perhaps maybe only one object is changed out for a new object. During this time, researchers actually measure the time that the rat spends with the old object compared to the new object. And since rats like novelty and they're attracted to novelty, they're going to want to spend more time with the object that was replaced. So ultimately, we can measure spatial learning and memory based on the amount of time that a rat prefers to spend with a new object compared to an old object. And several studies utilizing paradigms such as the novel object recognition task or the object location memory task have demonstrated that estradiol enhances these forms of memory formation. So as I mentioned previously, in these experiments, the rats are trained to run a maze perhaps, or to remember objects and their places. So if you want to learn more about these particular behavioral paradigms, you can also reach out to me, but I advise you to explore the novel object recognition paradigm or the object location memory task. So now let's talk about estradiol and memory during these tasks. So for example, in one study, estradiol was administered shortly before or after the training period during the novel object recognition task. So remember, the novel object recognition task would require an animal to explore a chamber with uninterrupted movement for about 10 minutes, and they would be able to explore two different objects in the chamber. They'd be put back in their home cage, and then hours later, perhaps 24 hours later, they'll be placed back into the chamber, but there will be a new object in place of the previous object. Then scientists measure the amount of time that the rat spends with the old object compared to the new object to test spatial memory. And it's interesting to note that if estradiol treatment was administered before or after training, it enhances their performance and allows them to recognize that a new object replaced the previous object. They'll likely spend more time with the object that was replaced. But if estrogen is not given within that 2 hours before or after training, this memory benefit associated with estradiol disappears. So thus, it's important to know that estrogen is actually facilitating memory, and it needs to be present near the time of the learning experience in order for the rats to have this memory benefit associated with estradiol specifically. So to compare this to human studies, this would mean that a human female would need to have estradiol present during learning and memory tasks to bolster those tasks. Estradiol has also been shown to have a protective effect on neurons that combat disease. So it's amazing to me when we think about estradiol and the role it plays in our mood, in our behavior, in our cognition, that it even plays an important role in helping in disease prevention. So, in some studies, scientists conduct in vitro experiments. This is when they can take cells, perhaps they'll take neonatal cells from a rat or mouse and grow them in culture. Cells are grown in petri dishes, and this is a great way for scientists to test direct cause and effect in cell lines to see how things like oxidative stress, which is really the accumulation of reactive oxygen species, can affect neurons. Now, cells are going to die in response to oxidative stress. So, for example, if you treat them with hydrogen peroxide or another reactive oxygen species, it will usually decrease cell viability and increase cell death. However, in some studies, scientists have demonstrated that if cells are treated with estradiol, they're protected against oxidative stress. So, estradiol can play a protective role in diseases associated with oxidative stress. And a key disease associated with oxidative stress is dementia and Alzheimer's disease. Ultimately, as you know, I study Alzheimer's disease, and I think hormone replacement therapy for Alzheimer's disease and even Parkinson's disease could be a really interesting therapeutic strategy. So during menopause, women have this huge drop in estrogen. And now you know that estrogen plays such an important role in learning and memory, dendritic spine density, and protection against oxidative stress. So thus, women don't have this important hormone that is going to provide protection for them. So thus, studies have actually started to investigate whether estrogen replacement therapy could potentially delay the onset of Alzheimer's disease, or even decrease tremors present in the neurodegenerative disease called Parkinson's disease. And so in the study, which I attached the link here if you want to go read it, this was published in the journal Alzheimer's and Dementia. They found that women who underwent menopausal hormonal therapy for 6 years or greater were 79% less likely to develop Alzheimer's disease and 77% less likely to develop any neurodegenerative diseases. Now, this research is absolutely incredible because it really does demonstrate that there is an association between estrogen and long-term health, in addition to the development of neurodegenerative diseases. So ultimately, this is newer research. There still needs to be a lot more research and studies done to look at the effects of hormone replacement therapy and disease prevention. I will note that there are other studies that have also found that hormone replacement therapy can actually increase the risk of certain cancers. So you do have to do your own research to see if this is something that you would want to partake in. But ultimately, hopefully we have more research that's contributing to this effort to see if hormone replacement therapy, such as estrogen replacement therapy, could be utilized to reduce the risk of Alzheimer's disease in women. And women are far more likely to get Alzheimer's disease. 2 out of 3 Alzheimer's disease patients are women. So ultimately, this research that's tailored to the study of female hormones, like estrogen, are really important. And menopause could be a way that women have this increased risk of Alzheimer's disease, and replacing that loss of estrogen could be potentially utilized as a therapeutic strategy. But again, more research needs to be conducted to understand the complex biological and psychological interactions. Thanks for listening, and I hope you guys have a great rest of your week.